Hello students, welcome to Grade 12 Biology Unit 1 Part 4. In this video we will talk about laboratory rules which must be followed when working with microbes. Before we start, make sure that you subscribe to this channel and like this video. Microorganisms Safety Guide. Bacteria are ubiquitous, and live within the human gut, and in every corner of our environment. We come in contact with bacteria on a daily basis. Hand washing is 99.9% effective at decontaminating us from bacteria that might reside on the skin. Therefore, when the proper safety precautions are taken, colonies of microorganisms can be safely isolated from homes, yards, gardens, etc.
The majority of microorganisms are non-pathogenic, but bacterial cultures or petri plates that contain any type of bacterial colonies should be treated with general safety precautions.
Here are 9 safe practices for the microbiology laboratory that you should follow when working with microorganisms. James, 2008.
1. Treat all microorganisms as potential pathogens. While the majority of microorganisms are not pathogenic to humans and have never been shown to cause illness, under unusual circumstances a few microorganisms that are not normally pathogenic can act as pathogens. Treat all microorganisms, especially unknown cultures, as if they were pathogenic. A student who has a compromised immune system or has had a recent extended illness should talk with his or her instructor before working in the microbiology laboratory.
2. Sterilize equipment and materials. All materials, media, tubes, plates, loops, needles, pipettes, and other items used for culturing microorganisms should be sterilized by autoclaving. Otherwise, use commercially sterilized products. Understand the operation and safe use of all equipment and materials needed for the laboratory.
3. Disinfect work areas before and after use. Use a disinfectant, such as a 10% bleach or 70% ethanol solution, to wipe down benches and work areas both before and after working with cultures. Also be aware of the possible dangers of the disinfectant, as 70% ethanol can catch fire around open flame or high heat sources. Bleach, if spilled, can ruin your clothing. Either alcohol or bleach can be dangerous if splashed in the eyes. You should know where the nearest eyewash station and sink are located.
4. Wash your hands. Use a disinfectant soap to wash your hands before and after working with microorganisms. Non-disinfectant soap will remove surface bacteria and can be used if disinfectant soap is not available. Gloves may be worn as extra protection.
5. Never pipette by mouth. Use pipette bulbs or pipetting devices for the aspiration and dispensing of liquid cultures.
6. Do not eat or drink in the lab, nor store food in areas where microorganisms are stored. Never eat or drink in the laboratory while working with microorganisms. Keep your fingers out of your mouth, and wash your hands before and after the laboratory activity. Cover any cuts in your hands with a bandage. Gloves may be worn as extra protection.
7. Label everything clearly. All cultures, chemicals, disinfectants, and media should be clearly and securely labeled with their names and dates. If they are hazardous, label them with proper warning and hazard information.
8. Autoclave or disinfect all waste material. All items to be discarded after a class, such as culture tubes, culture plates, swabs, toothpicks, wipes, disposable transfer needles, and gloves, should be placed in a biohazard autoclave bag and autoclaved 30 to 40 minutes at 121 degrees Celsius at 20 pounds of pressure. If no autoclave is available and you are not working with pathogens, the materials can be covered with a 10% bleach solution and allowed to soak for at least 1 to 2 hours.
9. Clean up spills with care. Cover any spills or broken culture tubes with a 70% ethanol or 10% bleach solution, then cover with paper towels. After allowing the spill to sit with the disinfectant for a short time, carefully clean up and place the materials in a biohazard autoclave bag to be autoclaved. Wash the area again with disinfectant. Never pick up glass fragments with your fingers or stick your fingers into the culture itself. Instead, use a brush and dustpan. If working with animal or plant pathogens, keep the area clear and notify your instructor. That is all for today. Make sure you subscribe to our channel. See you in the next video. Bye.